Hi guys, it's your science teacher here back with another video. This time I'm going to go through the whole of biology paper 1. This is the expected questions for your AQA exam. Now what I've done is I've had a look at the document that AQA have released about what topics they've included in this year's paper and what they have said will not be on the paper. So I've put together a few slides going through what they have said is definitely going to be on the paper. Now, what I would say is that this video will only briefly go over these topics. If you want to look at them in more detail, please watch the end of topic videos. But without further ado, let's get started with today's video. So you might be wondering where I've got this information from. I'm not a mind reader, but I have visited the AQA website and they have said what is going to be on the higher paper and foundation paper. This video is specifically aimed at triple science students, so those people who are doing single biology. And you can see here that it said that, for example, they will cover cell structure, transport in cells, animal tissues, communicable diseases on both higher and foundation. So I'm going to talk about them in this video. I'm also going to go over monoclonal antibodies for those higher tier students, and I will be doing the plant tissues as well for them. And I will also go at the end of the video for over photosynthesis for those doing foundation. So please do stick around for that. We're going to start today's video off by looking at the required practicals that it says is going to be included on your GCSE biology paper 1. And the first practical it says that's definitely going to be on the paper is microscopes and observing cells. And in specific, we use light microscopes in our GCSE science syllabus. And how do we set it up? Well, we need to know what our microscope actually looks like and the different parts of our microscope. And we look through the eyepiece that's on the top of our light microscope. And this does provide some of the magnification. The other part of the microscope that provides magnification is our objective lens. You can actually calculate how much magnification you are going to have by timesing the objective lens magnification by the eyepiece magnification. For example, if this was times 5 and this was times 10, your overall magnification would be times 50. Underneath our objective lens is our stage, and on our stage we put our lens with our sample on. Then we have two focusing knobs on the side of our microscope. There is the coarse focusing knob and the fine focusing knob. The role of the coarse focusing knob is to bring the specimen into approximate focus, and then we use the fine focusing knob to sharpen the quality of the image. You don't usually use the fine focusing knob till after you've used the coarse focusing knob in order to see your sample. Now, if we use our microscope, we can find out the size of our sample by using the magnification equation. And to work out the magnification or work out the actual size, we use this calculation triangle over here. And if we look at the image size divided by the magnification will give you the actual size of your image. For example, if your cell has a width of 4 millimeters under a microscope and it has been magnified 100 times, you could work out the actual size of your cell by doing 4 divided by 100. And because you are using incredibly small numbers in this topic, it's also important to remember your prefixes. You have milli, which is times 10 to the minus 3. You have micro, which is times 10 to the minus 6. You also have nano, which is times 10 to the minus 9. And if it gets small enough, you might even get picometers, which is times 10 to the minus 12. If we also look at how to set up our slide in order to put it under the microscope, here I've got my glass slide and I'm going to place my sample on it. Now, once my sample is there, that could be an onion cell, it could be a cheek cell. What you then do is you're going to put some indicator solution. Usually for plant cells, you will use the solution iodine on top of it. So once you've got your iodine on top of it, what you do is you lower a cover slip down on top of your slide, and you lower it really slowly. The reason why you're going to lower it slowly is that you don't want there to be air bubbles underneath, and that's a common problem with looking under things under a light microscope. Is sometimes you're looking at air bubbles, and the reason why you get air bubbles is because you didn't lower the cover slip slowly. The next required practical that you need to know off by heart is your food tests. And what we do in these food tests is we test for different nutrients that are in food, and if we get a positive test, we know that nutrient is present. The first one we are going to look at is the food test for sugar. And to test for sugar, we add Benedict's reagent. And we need to remember with this food test, actually, that we often need to heat in a water bath as well. It needs a bit of heat for you to actually see the colour change. I'll go through all the positive result tests at the end after the reagents for all of the different tests. So the next one is testing for starch, and we test for starch with iodine solution. And after starch, we have protein, which we test for with burette solution. And then finally, we have fats, lipids. And what we do is we add ethanol and water. Now, if we do the sugar test and we add our Benedicts and we heat, what we observe is we observe blue going to red. This is for a positive test, meaning that it contains a lot of sugar. Now, sometimes you can observe it going yellow if it doesn't contain lots and lots of sugar. Now, the starch test will go from a brown colour. Some people do describe it as yellowy black, but I prefer to describe it as a browny colour, and it goes black if you have starch present. For proteins, once you add the burette solution, burette is also blue, and if you get a positive result for this test, that burette solution will turn purple. And finally, for the fats and lipids tests, what you get if you get a positive result is you get a white emulsion. Now, you're just going to have to learn these tests off by heart, because it could come up with either of these food tests in your exam. What you also need to remember is if you have a solid sample, what you'd want to do is you'd want to crush it up with your pestle and mortar, and you'd want to add some water to it in order to make the nutrient that you want dissolve in that water, so it will react with the reagent. The last practical on your biology paper 1 is the Osmosis Practical, in which you probably use potato chips, but it could be any vegetable coming up in your exam, as previous students know they use carrots in an AQA exam question. But the key principle behind this practical is you get 3 vegetables and you cut bits up and you put them in 3 different solutions. Now here we have distilled water, which is my hypotonic solution. Hypotonic means that the solute is in less concentration. Now the solute could be salt or it could be sugar. It doesn't really matter. I've used a salt solution here. And here I've got my isotonic solution, in which my solute and solvent are in similar concentrations, and I have my hypertonic solution, in which my solute is in a high concentration, as shown by these 3 images. This one over here is my hypotonic, this one is my isotonic, and here is my hypertonic solution. Now what you do, now you've got your 3 solutions, is you leave your vegetable inside them solutions and you weigh your vegetable before and after, and what you need to do to calculate the percentage change in mass is you do its new mass minus the old mass divided by the old mass times it by 100 to turn it into a percent. And what you should observe is in this hypotonic solution, where there is more solvent particles, the potatoes chips mass should increase. If the concentration of salt or solute, which could be a sugar as well, is the same, what you should see is that it stays about the same mass. And what you should see in our hypertonic solution is that the mass decreases. Now, there's not really much more to this, just the key principle of osmosis to why this happens. Basically, if the concentration of water outside the potato is greater than that inside, then it will move into the cells through the partially permeable membrane, and that is what causes the increase in mass that you see in the hypotonic solution. Whereas in the hypertonic solution, the concentration of water inside the potato chip or any vegetable it could be is greater, so it's going to move outside of the cells into the solution, and therefore its mass will decrease. Now we're going to look at cells and cell structure, because that is another one of the topics that it says it will cover on your biology paper 1 exam. And let's first look at 2 different types of cells. We have prokaryotic cells, which are single cellular, and we have eukaryotic cells that make up multicellular organisms. So eukaryotic cells can be found in animals as well as plants, and over here we just have a generic animal cell. And if we have a look at some of these key parts in the animal cell, in this black blob in the center of my cell, this is my nucleus, which stores the DNA. Around the outside you have the cell membrane. And this is like your bouncer of your cell. It lets things diffuse in and out of your cell. The jelly-like substance in which you find all of your cell is the cytoplasm, and this is where all the chemical reactions take place as well. Then we also have something new to key stage 4 that's not in key stage 3 is your mitochondria, and this is the site of respiration. Then we also have, you can see dotted around, ribosomes. And ribosomes is where protein synthesis happens in your cells. Another eukaryotic cell over here is just a generic plant cell, and they have a few other organelles that make up their cells, and we're just going to look at them ones in this video because, like I said, we're not going to deep dive into everything in this video. One of the extra features is the cell wall around the outside, and this just provides extra rigidity to the structure. And we also have our chloroplast, and our chloroplast is where photosynthesis occurs. This is the site of photosynthesis. The chloroplast contain a substance called chlorophyll as well, which absorbs sunlight, and this catalyzes the photosynthesis reaction. And then also we have a vacuole as well, and the vacuole's job is to store the products of photosynthesis. It stores like sap in order to store the products of photosynthesis. Down here I have the other type of cell, which is a prokaryotic cell. Prokaryotic cells can include any unicellular organism such as bacteria, and here we have some of the main features that you can see on a prokaryotic cell. For example, a lot of them have flagellums. This is how they move around. Flagellum is just another name for a tail. They don't have a nucleus. This is one of the main ways you can distinguish between them. They just have strands of DNA. A small spherical strand of DNA can be called a plasmid, and here as well we also have some more DNA. So there's the two ways in which it stores its genetic information. We also have, just like with our regular cells, we have cytoplasm, which is the jelly-like substance in which the chemical reactions take place, ribosomes for protein synthesis. Now, most have cell walls in order to stay rigid, and they all will have this membrane here so that substances can diffuse in and out of that cell. We're now going to focus on how things move in and out of your cell, and the first process for transport that we're going to look at is diffusion. And diffusion is a passive process, meaning that it doesn't require energy. And what happens in diffusion is particles move from a high concentration to a low concentration. Now, if I look at this cell down here, you can see that there are particles on the outside. This could represent oxygen, and these oxygen particles are going to move into my cell through my cell membrane due to the concentration gradient. That means that they're going to move in because there's more on the outside than the inside. Now, carbon dioxide does the opposite to oxygen. Carbon dioxide is in a greater concentration in your cell, so will move out of your cell. Now, there's a few things that you can do to increase the rate of diffusion. What you can do is you can increase your concentration gradient. That means have a greater number of particles on the outside than the inside so that they will move into your cell. You can increase the temperature. The higher the temperature, the faster particles move around. So this is why diffusion happens at greater speeds. And you could increase the surface area. The more surface that it's touching, the more likely that you're going to get diffusion into that cell. If we look at some key areas in the body where diffusion happens, we could look at alveoli in the lungs, and the alveoli in the lungs are adapted for diffusion by having an incredibly large surface area, and also they have a really high blood supply close to the alveoli, so they've got more blood there, so you're going to get more diffusion happening. Also, you can have thin cell membranes, and the thinner the cell membrane, the more oxygen can diffuse into our blood. Another area of the body where you have A lot of diffusion is in the villi, which are found in the small intestine, and the villi are there to allow nutrients to diffuse into our blood. And exactly the same, they have incredibly thin cell membranes as well as having high blood supplies and large surface areas. So you've got to think about those things when it's asking for adaptations in your body. You've got to be thinking thin cell membrane, large surface area, and also a high blood supply. Osmosis is another cell transport method, and it is also a passive process from high concentration to low concentration. However, this time osmosis only happens with water, and it happens through a partially permeable membrane. Now, if we have a look here, I've got lots of water on this side of my screen here, but I don't have any water on this side of my screen here. So what's going to happen is the water is going to move over until they are in equal concentrations on both sides. Now, just like with diffusion, it doesn't require any energy, and also I can increase the rate of my osmosis by increasing the temperature, increasing the surface area, and increasing the concentration gradient. The last cell transport method to look at is active transport. And active transport is different to osmosis and diffusion because in active transport, instead of going from a high to low concentration, we're going from a low to a high concentration. And because we're doing this, it requires a lot of energy. Now, because it requires energy, cells only do this for really important things that it really wants. For example, active transport happens in root hair cells in order to get some key minerals that the plant needs in order to grow. Now, root hair cells have adaptations in order to make sure that they can carry out active transport. They contain lots of mitochondria in order to have the energy to be able to get enough minerals. In this picture, you can actually see the concentration on the outside of the cell is less than inside the cell. However, they are still being transported into the cell in order to provide enough minerals. Going from cells ties into our next topic really well because we are going to look at organization. And we call cells that work together tissues. An example of some tissue would be muscle tissue. Now, muscle cells work together in a lot of organs to make up tissue. Organs is groups of tissues and other cells that work together for a specific function. For example, some organs in the body could be the liver, the brain, the heart, but all they are is tissues that work together for a specific function. Then organs that work together make up organ systems. Some examples of some organ systems could be the circulatory system, which is how blood gets pumped around our body. And another example could be the skeletal system, and that's what makes up our bones. Now, when we have organ systems working together, that makes an organism, and organism just means living. So you are an example of an organism. We're going to look at an example of a plant organ first, and the plant organ that we're going to look at is a leaf. Now, a leaf contains many different cell tissue working together, and we'll just have a look at some of them tissues. On the outside, we have the waxy cuticle layer, and this is for protection. And underneath, we also have the upper epidermis, and the upper epidermis is also for rigidity of your leaf. Then underneath the upper epidermis, we have the palisade mesophyll layer, and the palisade mesophyll layer is where photosynthesis occurs. The reason why it's positioned here is so that it can get the most sunlight as possible, and also it's still close to the spongy mesophyll underneath, so that gas exchange can take place and it can get enough carbon dioxide and get rid of some of the oxygen that it stores. Then if we look underneath our spongy mesophyll, we have the lower epidermis layer, which is another protective layer and for rigidity. And we also have our stomata, which lets gases come in and out. So that's why they can open and close, and they get open and closed by these guard cells here. Now, it's important to remember that plants need to keep hold of a lot of the water that they store. So the stomata don't stay open at all times. They will only really stay open during daytime, and at nighttime they close in order so that they don't lose water outside of really photosynthesizing hours. We also have over here the xylem, and the xylem transports water up from the roots, and we also have the phloem, and the phloem is different to the xylem. The xylem is made up of dead cells called lining, whereas the phloem contains lots of mitochondria and live cells because of the fact that it transports products of photosynthesis around your plant, and for that reason it needs to work both ways. So it takes things up from the roots, like some minerals, and but it might need to take some things down, for example some of the products from photosynthesis. And that's why it's not a passive process. It actually contains sieve cells and it requires live cells in there. So the leaf is actually a lot more complex than it first looks, but once we learn some of the key parts of our leaf, they are pretty easy to describe. We're now going to look at two human organs, and the first one we're going to look at is the heart. And the heart is quite special because it pumps the blood all around our body, and in humans we have what is called a double circulatory system. This means that the blood enters the heart twice in one full loop. Now, blood enters your heart through the main vein in your body, and that is called the vena cava, and it enters into your right atrium. Now, we call it your right atrium because it's right as if you imagine you are in the page. Now, underneath our right atrium, we have our right ventricle. And after the blood has been in the right ventricle, it gets pumped to the lungs. And it gets pumped to the lungs so that it can become oxygenated. And we call this blood vessel our pulmonary artery. Now, a good way to remember this is pulmonary is another name for lungs. So once it's gone to the lungs, it comes back to the heart through the pulmonary vein. And once it's re-entered our heart, it goes through the left atrium and the left ventricle, and it will go out of our heart and all around the body through the aorta, and the aorta goes all over the body. This blood is at really high pressure. This is why the left hand side of my heart is much thicker, because it's pumping blood all around the body. Now, when your blood arrives at your lungs have some pretty cool adaptations to allow as much diffusion to take place. Now, what happens is that air arrives through your trachea, which is known as your windpipe, and it splits in two in the bronchi, and it travels down the bronchi to our bronchioles. And at the end of our bronchioles, we have tiny air sacs known as alveoli, and this is where our gas exchange takes place. And your lungs will have millions and millions of alveoli to maximize your gas exchange. And this whole process of getting your blood around the body and getting it oxygenated is called the circulatory system. We're going to have a look at a different organ system in our body now, and that is the digestive system. And digestive system starts at your mouth, and your mouth chews up your food before it travels down your esophagus. And what happens in your esophagus is we start adding some enzymes before it heads all the way down to your stomach. And your stomach is adapted to start breaking your food down by having gastric acid. And gastric acid is really low pH acid. It's actually hydrochloric acid inside your stomach. And by your stomach you have your liver, and your liver produces bile, which neutralizes some of your stomach acid, and it also helps to emulsify some fats. Now, after your food has traveled through your stomach, it goes to the small intestine, and your small intestine is where most of your digestion takes place. As I mentioned earlier, it contains villi, and villi basically remove all them nutrients from your food and transfer it into your bloodstream. Now, after you have done this, your small intestine are incredibly long, so you've got a lot of the good stuff out of your food. It travels to your large intestine before your food is, the waste from your food is removed through your anus. Now, it can take over 8 hours for your food to pass all the way from your mouth through your small intestine. Now, on the way, it's going to encounter different enzymes. And the way enzymes work is they are specific to the nutrient that they work on. For example, proteins are broken down by the enzyme protease into amino acids. We also have lipids and fats, which are broken down by the enzyme lipase, and they're broken down into fatty acids and glycerol. And we also have starch, which is broken down by amylase into glucose. Now, the way these enzymes work is by something called the lock and key mechanism. Basically, it just means that that each enzyme is specific to that nutrient group, and they bind together in an enzyme substrate complex before breaking off and producing smaller products. Now, it's really important to remember as well, enzymes have specific pHs and temperatures in which they work best at. So depending on where they are found in the body depends which pH and which temperature they work best at. If the pH is too high or too low for that enzyme, they will denature, and same with the temperature as well. At too high of a temperature, your enzymes start to denature, meaning the active site changes and they will no longer fit the substrate. We're now going to look at communicable diseases. And communicable diseases are diseases that can be passed from one person to another person. And we say that communicable diseases are spread by pathogens. And we're going to look at 4 examples of pathogens. Pathogens are just disease-causing microorganisms, and the first one we're going to look at is bacteria. An example of a bacteria infection could be gonorrhea, and gonorrhea causes you to get pus, in your sexual areas, and it can be quite painful and cause pain while urinating. And another example of bacterial disease is Salmonella. And Salmonella is caused when you don't cook your food properly. It often breeds on raw foods, and when you ingest it, it basically multiplies really quickly in your body. Now, we treat bacterial infections with antibiotics. Basically, antibiotics will kill the bacteria and stop it from reproducing in your body. Another example of a pathogen is viruses. And viruses are very different to bacteria. They're incredibly small, and basically with viruses, they can't reproduce by themselves like bacteria. So what they have to do is they get inside a host cell, and they get inside the nucleus of your host cell, and when the cell divides, the virus divides as well. Now, viruses can't be treated by antibiotics. What they have to be treated by is you will use painkillers and wait for the symptoms to go away. Some examples of viral diseases are HIV, which attacks your immune system. When your immune system is weak, you are often susceptible to other diseases, and that's what actually causes you to get incredibly ill. Now, HIV is a sexually transmitted disease, just like gonorrhea. So the way to protect yourself from it is to wear a condom when having sexual intercourse. Another example of viral disease is measles, and measles causes itchy spots, and it's very unpleasant to have. It can be spread through direct contact, so through touching someone who has the disease. Plants can also get viral diseases, and an example is tobacco mosaic virus. And with plants disease, they are quite hard to treat because of the fact you have to weigh up whether you chop off just the infected area, or you decide to just burn the whole crop because it's just easier to stop it spreading to everything. Another example of another pathogen is fungi. And an example of a fungal disease is athlete's foot. And athlete's foot causes you to get scabs on your feet. It's quite painful at times. It basically breeds really well in the moist, warm environment inside shoes, and it gets spread a lot in sports teams' locker rooms and around swimming pools. Other examples of fungal diseases could be in plants as well. An example would be rose black spot. And like TMV, really, what it does is it stops the plant's ability to photosynthesize. And because of this, the plant can no longer produce all the products from photosynthesis, and it ends up just not being able to grow and dying. Another example of a pathogen is a protist. An example is malaria, and malaria is carried by the vector, which is a mosquito. Now, malaria can make you incredibly ill. It can cause you to be coughing, lack of air, and can eventually lead to death if it's not treated. Now, when you get a question about communicable diseases, what you need to do is basically identify whether it's viral, bacterial, or fungal, and then look at ways of treating that. For example, if you have a bacterial disease, you know you're going to treat it with antibiotics. Viral diseases, you're going to treat with painkillers and definitely not antibiotics. Fungal diseases, you can use fungal creams. And protists, you can also use medicines in order to treat that as well. However, the main protection against the malaria parasite actually is prevention. So we use mosquito nets in order to stop the mosquitoes from biting you. We're now going to look at monoclonal antibodies. And what we do to create monoclonal antibodies is we inject a mouse with antigens of either a disease or a hormone that we want to detect. And what the mouse will do is the mouse will produce antibodies against that antigen. And what we then do is we take these antibodies out of our mouse and we combine them with tumor cells. And the reason why we do this is because tumor cells grow incredibly fast. So these antibodies are going to reproduce quickly as well. So once we have lots of antibodies, we can then take these cells and we can use them for whatever purpose that may be. One example of where we have used monoclonal antibodies is in pregnancy detectors. What happens in a pregnancy detector is the hormone that you produce when you are pregnant, basically the antibody will attack it when you wee onto that pregnancy test, and therefore you'll get a line appear on your pregnancy test, and that's due to these monoclonal antibodies attacking that hormone, and that's what produces the line. Now, monoclonal antibodies are thought to have incredibly useful future uses because of the fact that they can do this and attack these antigens. It's been thought that maybe they could be used to treat some cancers because they can attack cancer cells, making them easier to detect or stop them cancers from growing. But the problem with monoclonal antibodies is sometimes they have been quite dangerous because of the fact that we use mice in order to collect a lot of the antibodies. Sometimes it doesn't fit too well with humans' bodies, and sometimes they get rejected and it can have some catastrophic consequences. So it's really important that we test out our monoclonal antibodies before bringing them to market. If you are doing higher tier, well done, you have finished today's video. I've done it a bit of a different way around today. I've done the higher tier first and foundation second because a lot of my higher tier students will complain and they always have to watch the end of the video and foundation don't. So today is a little bit different. Foundation tier, you have a little bit left to go. We're going to look at the last topic on your paper, which is photosynthesis. Now, photosynthesis is how plants make their own food, and water and carbon dioxide are combined to make glucose and oxygen. And we've already looked at a cross-section of a leaf, but I thought it's always better to see the actual photo of what a leaf looks like on the inside. So here you can see the spongy mesophyll layers, you can see the xylem and the phloem, and you can see that palisade mesophyll layer underneath your waxy cuticle, and this is where all of that beautiful photosynthesis takes place. Now, it's incredibly important that your plants make enough glucose. Some of that is used to grow. Some of it will be stored as starch. Some of it is converted to cellulose, which is used to make cell walls, which is incredibly important to provide rigidity to your plant cells. So your plants are completely perfectly adapted to photosynthesize all the way from the roots all the way up to your leaves. Now, photosynthesis can be affected by a few things. These are called limiting factors. The first one we will look at is light intensity. The stronger the light, the faster the rate of photosynthesis up until a certain point. And at this certain point, another limiting factor is basically limiting the amount that it will photosynthesize. Now, I could make this plant photosynthesize more, but I would have to increase one of the other limiting factors, for example, temperature or carbon dioxide concentration. The temperature graph for photosynthesis is a little bit different. because of the fact at really high temperatures enzymes denature. This means that active site changes, and therefore your enzyme will no longer be able to work, and photosynthesis basically just won't happen. Carbon dioxide concentration is very similar to light intensity. At a certain carbon dioxide concentration, that is the highest level of photosynthesis, unless you change another one of the factors. Now, for farmers, it's incredibly important to consider all of these limiting factors. That's why they design greenhouses in certain ways to maximize light intensity and reach the optimum temperature and carbon dioxide concentration for quick growth. Now, that is the end of the video, unfortunately. Thank you for sticking around all the way to the end. Remember, if you did like today's video, drop it a like and subscribe to the channel.